All right, what's up, guys? So I'm just going to redo the part 1 video for the first essay prompt talking about depolarization and muscle contraction. So let's get into it. All right, so we've got our heart. I don't know why I said it like that. Our heart right here kind of looks like a small little potato, but we're going to go ahead and get into it. So let's first define what a depolarization signal is. So a depolarization signal is simply just a shift in electrical distribution of a cell membrane. And depolarization signals are important because contraction of a muscle is going to follow that electrical depolarization signal. So we require the depolarization signal to stimulate a contraction, if that makes sense. And so when we're talking about depolarization, specifically when it pertains to the heart, we're going to really be looking at this anatomical structure called the SA node. So the SA node is kind of the pacemaker of the heart. In other words, it has inherent automaticity. So it doesn't require another structure to initiate this electrical signal. It actually spontaneously depolarizes all by itself, which is really cool, and sends kind of that electrical signal to the AV node, which then passes it to right and left bundle branches and through the Purkinje fibers so that the whole heart receives this electrical signal, so then contraction can occur. And so I have these other sites listed. I have the AV node and then the left and right bundle branches and the Purkinje fibers, which I had just mentioned. And the reason that these are important is because these are also sites of inherent automaticity. Well, how does that work, Peyton? Well, there's something called SA node overdrive. So what's unique about the SA node is that it spontaneously depolarizes quicker than the rest of the sites of inherent automaticity, meaning that it actually suppresses the other sites. But the reason that this is important is because if the SA node fails, the heart has 3 other rescue mechanisms to which it can continue to fire these electrical impulses so that the heart can continue to function, which really stresses the importance of these electrical signals. So what do I mean by this? If the SA node malfunctions or fails, then it's the AV node's responsibility to pick up the slack and begin initiating those electrical signals through inherent automaticity. The AV node is the second fastest foci for initiating these electrical signals. But then if the AV node fails as well, and the SA node fail, then these right and left bundle branches will be responsible for initiating that electrical signal. But these are slower than the previous. These are slower than the AV node. But then if the left and right bundle branches fail as well, then the Purkinje fibers are going to take over. And these are going to be kind of the heart's last rescue mechanism to really send these electrical signals throughout the course of the heart. Purkinje fibers kind of being the slowest of all of them. Okay, so we have these electrical impulses traveling through the heart, but we have something known as an EKG, which can actually measure these electrical impulses through the heart, and they have kind of different waves to symbolize where the electrical signal is traveling through the anatomical structures of the heart. So let's talk about that. So this little bump is called a P wave. It's denoted by this little bump, and this P wave represents atrial depolarization. So that's depolarization traveling through the atriums, okay? Followed by this little plateau, which is called AV nodal delay, which occurs so that blood can fill into the left ventricles from that atrial depolarization and contraction. So it's important to note that depolarization is not the same thing as contraction, but contraction typically follows a depolarization signal. But depolarization initiates that contraction. Okay, so we have the AV nodal delay, which follows depolarization, which follows contraction, so that we can have that blood, max amount of blood kind of filling into those ventricles. Then we have something called the QRS complex. And so the QRS complex is hugely important and represents ventricular depolarization. And the reason this depolarization looks different from the atrial depolarization is because depolarization through that ventricle has to be very forceful, because that ventricle, specifically the left ventricle, is responsible for pumping blood to the entire body. Yes, that small little chamber of your heart is what is responsible for pumping blood systemically, which is crazy. So a strong depolarization signal needs to initiate so that we can have a strong contraction, right? And so you can see kind of Q to R, the signal is traveling through these bundle branches, okay, these bundle branches. And then R to S, Purkinje fibers, are these kind of traveling on the outside of the heart, or outside of the ventricles, so that we can kind of have that super forceful contraction. And I kind of drew this a little wrong. S is actually supposed to dip down a little bit further, but that's on me. Hidden within this QRS complex is also atrial repolarization. And this is the atria's kind of attempt to reset itself so that it can receive another depolarization signal to fire again, so that contraction can follow and blood can flow into the ventricles. Then after that, we have this T wave, which simply just denotes ventricular repolarization. The concept is the same with ventricular repolarization as it is with atrial repolarization. Just the ventricles kind of hitting the reset button so then they can receive that depolarization signal all over again. Awesome. So now we have these awesome graphs. So these graphs are known as action potential graphs. Okay, you may be asking yourself, like, what is a action potential? An action potential simply just measures the fluctuation in ions across a cell membrane. That is literally all it denotes. And so I really like to think of these graphs as literally almost an exchange that is being performed. And I'll talk more about that once we get into the graphs specifically. But why do these graphs look differently? So let's focus on this pacemaker action potential first. So at the beginning of this, I said that the SA node is kind of the pacemaker of the heart, right? And you'll notice how this pacemaker cell doesn't really have a resting membrane potential. It doesn't have a true resting membrane potential because it's always kind of upticking and firing that depolarization signal, because these pacemaker cells are at sites of inherent automaticity. Okay, so at the SA node, at the AV node, at the right and left bundle branches, and at the Purkinje fibers, because they can spontaneously depolarize, meaning that they don't have a resting membrane potential, which is really, really cool, right? And so what in the world do I mean by it's kind of a transferring of ions? Well, look at this with me. So look at this kind of graph. So right here, we kind of have a slow leak of calcium into these pacemaker cells until it hits this dotted line threshold, and then it shoots really high up. And so we have a high influx of calcium intracellularly, therefore making the cell a lot more positive because calcium is a positive ion. But I like to think of these ions almost like currency, okay? So you kind of envision this analogy. All right, so if I'm a person and there's another imaginary person in front of me, pretend that that person is the cell, okay? I like to think of it like, okay, I'm giving this cell currency. I'm giving this cell calcium, and it's becoming wealthier. It's becoming more positive. So that means that it's depolarizing, okay? I've given this cell calcium. I've given it money. It's becoming more positive. It's becoming wealthier. But then, the cell feels really bad, and so the cell kind of feels guilty, and so the cell actually tries to pay me back. And so the cell tries to pay me back with not calcium, but potassium. And so that's why we have this repolarization, is because the cell feels bad and is trying to give me money in exchange. And potassium is a positive ion as well. And so this cell is actually becoming poorer because it's paying me back. So it's becoming more negative. right? So then it can restart this entire process again and send that electrical signal and depolarize. So why is this pacemaker action potential important? Well, because this pacemaker action potential is what sends the electrical signal to these cardiac myocytes. And we're going to talk more about these cardiac myocytes here in just a second. But these cardiac myocytes are unique in the sense that they have something called intercalated discs. I like to think of these intercalated discs as almost bridges connecting each and every cell to each other so that that electrical signal can pass freely between cell to cell. It's like that signal taking those bridges to the next cell, then it's going to take that bridge to the next cell, then take that bridge to the next cell, right? And so these pacemaker cells initiate that signal, which then travel to these cardiac myocytes. And so the idea of this ion transfer is the exact same in these myocytes as it is in these pacemaker cells, but these are not at sites of inherent automaticity, if that makes sense. So what's happening in this cardiac myocyte? So in this cardiac myocyte, we do have a resting membrane potential, again, because these myocytes are not able to spontaneously depolarize. And so we're specifically looking at the ventricular myocytes here. So think these two bottom chambers here. So ventricular myocyte. So it has a resting membrane potential because it cannot spontaneously depolarize. But once it receives a signal from this pacemaker cell, then sodium is going to begin leaking into the cell, causing depolarization. So I'm giving the cell money. I'm giving the cell sodium. Therefore, it becomes more positive. But then the cell again begins to feel really bad. And so it begins to pay me back with potassium. And so the cell becomes a little bit more poor. So it's becoming a little bit more negative. But then I say, no, I insist, have my money. And so I give the cell a whole lot of calcium now, which denotes this plateau, right? So now there's an equal exchange. There's an equal influx of calcium and an equal efflux of potassium. I'm giving the cell calcium, but he's giving me potassium in return. And so we kind of have this plateau where he's not losing money and he's not really gaining money either. But then, unfortunately, I run out of money. But he somehow had some savings, and so he's continuing to give me potassium, and so potassium is leaving out of the cell. Therefore, he becomes poor. He becomes more negative, therefore repolarizing until it reaches this resting membrane potential. So then the cell can receive this signal from the pacemaker cells to then shoot again. Okay, why is this entire process even important, right? Why is electrical signal, why is depolarization signals initiated by these action potentials important in the heart, right? Well, because here's the thing. The SA node initiates this action potential, causing a depolarization signal throughout the course of the heart, which we can measure through this EKG. Right? And these are the action potentials that occur to initiate that depolarization signal. This whole process has to happen to send a calcium signal to the cardiac myocytes. This calcium signal is hugely important because this is how calcium is going to enter these myocytes. So look here with me. Cardiac means heart. Myo means muscle, cyte means cell. So we're looking at a heart muscle cell, okay? And look here. So the really cool thing here is you see the beginning of the kind of this yellow squiggly line? That is the calcium signal that has been given to this cardiac myocyte because the SA node that we can measure through the EKG has been initiated by these action potentials of the pacemaker cells, which then stimulate the cardiac myocytes to depolarize, which send this calcium signal across the surface of the cell. So you see how all of those things connect up until this point. And we're going to talk about muscle contraction in just a second. And so I know this picture is really big and confusing, but we're going to break it down. So let's focus on this yellow squiggly line first. So this yellow squiggly line is this calcium signal traveling across the surface of this cell, but then it hits kind of this dip. Okay, I like to think of this dip kind of like the Mariana Trench. Okay, so the signal dips deep into the Mariana Trench, deep within the muscle cell, so then it can initiate this process of contraction through something known as the sliding filament theory. So that signal needs to go deep within the cell to initiate this. But look at this. The signal continues across the surface of the cell until it reaches intercalated discs. So these were the bridges that I was just talking about, right? So I didn't have enough space on this page, but I could have drawn another cardiac myocyte connected through these intercalated discs, and that calcium signal would have then passed from these intercalated discs onto the surface of the other myocyte, reaching deep into that T-tubule or Mariana Trench, therefore initiating this contraction process as well, which is super, super cool. So those intercalated discs act like a bridge. Okay, why is this calcium signal important? Well, the calcium signal is picked up by these L-type calcium channels, which then stimulate the feet of the ryanodine receptors of the sarcoplasmic reticulum so that it can release calcium and calcium can come into the cell, okay? Now, calcium is quite crucial here. So we have something called the sliding filament theory, and this is how muscle contraction is actually initiated, and the ion calcium plays a crucial role in actually initiating this contraction process. So we have some contractile proteins known as myosin and actin. And think of myosin almost like your hand. It's trying to grab on to something. It's trying to grab on to actin. But there's something called a troponin-tropomyosin complex, which is blocking the binding sites of actin so that myosin is unable to grab it. So what happens is this calcium that was released from the sarcoplasmic reticulum, because that calcium signal was sensed by the L-type calcium channel receptors and passed it on to the feet of the ryanodine receptors, is now going to bind to this troponin C molecule, this little red circle. And so this troponin C molecule is going to be bound with calcium, which then allows the troponin-tropomyosin complex to shift off of actin so that myosin can actually grab onto this actin thin filament binding site, which is hugely important. And so troponin I is kind of what is connected to this tropomyosin. And troponin T is what's kind of in the way of this binding site. And so when calcium binds to this troponin C, this tropomyosin troponin complex can shift off of actin, and now there's an available spot for this myosin to grab on to actin. So myosin is the thick filament in this theory, while actin is the thin filament of this theory. Why is this theory so important? Here's the reason. This is how muscle contraction is actually performed, okay? So when calcium binds to troponin C, allowing the troponin-tropomyosin complex to shift off of actin, myosin can now grab actin. So think of it like my hand has grabbed a rope, okay? Now ATP hydrolyzes on this myosin, hydrolyzes into inorganic phosphate and ADP. When inorganic phosphate falls off of myosin, I pull this rope or I pull this actin 50%. Then once ADP falls off, I pull this rope or I pull this actin the rest of the 50%. And then ATP will bind to that myosin again. And so then I let go of that rope. Okay, why is this pulling action so important? Well, this is exactly what happens during contraction, okay? These muscle fibers, these contractile proteins are actually pulling the muscle fibers towards each other, and the muscle is actually able to shorten, okay? So again, that sliding filament theory, it's like pulling on a rope. I'm pulling it, okay? Think of like these fingers as pulling these, okay? It's pulling it, it's pulling it, it's pulling it, it's pulling it. Because when we contract our muscle, look at my bicep. My bicep is shortening, right? It's shortening towards the center of the middle, okay? Towards the center of the muscle, rather. It's shortening, right? And that's because of that sliding filament theory. It's pulling, that myosin is pulling that actin to be able to shorten that muscle so then I can perform elbow flexion, which is really, really cool. Okay, so myosin, again, pulling actin so I can shorten my muscle. Myosin pulling actin so I can shorten my muscle. Myosin pulling actin so I can shorten my muscle. So that's how important myosin and actin is. So then we have another molecule known as SERCA. So SERCA kind of acts as a vacuum that will reuptake this calcium and put it back into the sarcoplasmic reticulum. So then we have more calcium in the sarcoplasmic reticulum to be released intracellularly. So then we can go through contraction again. Phospholaminin can inhibit the activity of SERCA though. Phospholaminin is kind of denoted by here. Okay, so we have all of that. That's muscle contraction. But here's the thing. What happens during exercise? Exercise, we enter kind of this sympathetic nervous system state. So sympathetic nervous system is pretty much our fight and flight, okay? Fight or flight. And the reason we go into sympathetic nervous system is to try and increase cardiac output through increasing our heart rate, but also increasing stroke volume by increasing contractility. So this is key. The sympathetic nervous system releases a hormone known as norepinephrine and epinephrine, okay, denoted by here. So when we are exercising, we're in a sympathetic nervous system state, where the SNS has a nerve called the accelerator nerve that will again release this epinephrine and norepinephrine. Within these cardiac myocytes, there's something known as a PK alpha molecule. That PK alpha molecule is right here. Epinephrine and norepinephrine will cause this PK alpha cascade within the cell to activate 6 unique mechanisms to increase inotropy or force of contraction for the heart. So then we can actually maintain that cardiac workload during exercise, which is freaking sick. All right, so sorry if it kind of cut off really awkwardly. My phone storage was full, and so the video just kind of cut off, and I've been trying to take this part of the video for a while, but my phone has just continued to die and my storage has been full, but we're back. And I just got a 10% warning, so we're gonna see if we can do this pretty fast. But we left off talking about PK alpha. So PK alpha is going to kind of stimulate 6 different cascades to increase the inotropy of the heart or force of contraction of the heart during exercise, because we're more in an SNS kind of state. So the first way PK alpha is going to do that is that it is going to increase calcium influx through the L-type calcium channels to kind of stimulate this whole cascade even more, so then we can have more intracellular calcium binding to that troponin C, shifting that complex so myosin can grab onto actin. We'll have more contractile proteins connecting to therefore increase that number and increase inotropy. The second way that PK alpha is going to work is that it's going to stimulate the sarcoplasmic reticulum to release even more calcium. This is going to function in the exact same way, because if we have more intracellular calcium, then there's more opportunity for that calcium to bind to a lot more troponin Cs, shifting those complexes, therefore meaning that there's more myosin and actin cross bridges that can happen to perform a more forceful contraction. Third way is that PK alpha is going to increase the affinity for calcium on the troponin Cs, so that troponin C is like, oh, I want more calcium, give me calcium, give me calcium. So then more, that connection between calcium and troponin C is going to happen a lot more often, shifting those complexes, allowing for more cross bridges to form, increasing inotropy. The fourth way PK alpha is going to increase inotropy is that it's going to increase the activity of the myosin ATPase molecule, allowing for a more efficient pooling or ratcheting, which will therefore increase inotropy as well. And then the fifth way PK alpha is going to increase inotropy is that it's going to increase circuit activity by phosphorylation of phospholaminin. So if we can vacuum up more calcium, that's more calcium in the sarcoplasmic reticulum that can then be released intracellularly to bind to troponin C, therefore increasing those amount of cross bridges. The last way PK alpha is going to work is that it's going to decrease calcium efflux out of the cell. Because then, again, if we're losing calcium intracellular, if there's more calcium going out of the cell, then that's less calcium that can bind to troponin C. So that's less complexes, tropomyosin and troponin complexes that are shifting off of actin, and less cross bridges that can be formed. So we want more intracellular calcium to maximize the amount of cross bridges to really take advantage of that sliding filament theory. So that is contraction, that is depolarization. Again, I'm just going to edit these two videos together, but I hope you guys enjoyed and I hope you guys learned something.